I'm Bielawski. I'm a rising junior at the University of Illinois studying math and physics. I'm here to talk about the project I did this summer, Collective Behavior Stochastic Kinetic Theory, as a part of the Trend REU program. I'm going to introduce what collective behavior and the model that we take, quorum sensing, is. We're going to talk about the kinetic formulation, and then we're going to add stochasticity to our system, which is adding randomness. So as background, collective models are interested in the behavior of many individual agents that are able to act upon each other. Agent can mean anything from bacteria, fish, birds, wildebeest. It's a very widely applicable formulation. Such models are typically a system of coupled differential equations, where for every single agent, we keep track of position and velocity. The model that we take is the Cucker-Smale model, which is characterized by this interaction term. Essentially, every single agent tries to align every other agent's velocity with their own, modified by some function dependent on the distance between them. We particularly apply this to bacterial motion. So we take a model of bacterial motion called quorum sensing, where we allow bacteria to communicate with each other and sense the position of all of the other bacteria with a chemical known as an autoinducer. And so we have to keep track of not only position and velocity of the bacteria, but cell activity and concentration of autoinducer around the bacteria. We just adopt a biological model, which says that our system of ODEs should be this. This looks like a lot of terms. It's not that complicated. The first one is just transport, and then we see 3 Cucker-Smale terms, and then everything else is effectively just damping. This is literally damping. This is damping where the force of the damping is dependent on the autoinducer concentration. This is damping where the vector that it's being damped around depends on the cell activity. So this is a nonlinear coupled system of differential equations. We absolutely do not expect to get explicit solutions. However, we're still interested in analyzing what properties solutions must have. And so one such property is flocking. Flocking is mathematically defined as this. Essentially, we just require all of the bacteria to remain close to each other and their velocities and cell activities to align for a long time, and the autoinducer concentration gradients to remain bounded. As a basic theorem that we prove/improve upon previous results, we prove under certain assumptions that you get any solution to the quorum sensing model will flock. All of the proofs are relatively simple. You define certain energy functionals, and it turns out that all of the interaction terms are dissipative, and then you can end up getting the results that you need. Next, we're going to talk about the kinetic formulation of quorum sensing. Keeping track of individual bacteria is cool and awesome and like a tractable question, but oftentimes we don't care what individual bacteria are doing. We care about what a density of bacteria are doing, where bacteria are clumped, where they are sparse. And so instead of considering discrete bacteria, we're going to consider a continuous density. Of course, the obvious question is, how do we let a continuous density time evolve while still respecting the discrete system? And one just does a calculation of this following like BBGKY hierarchy. It's a very long and boring calculation, and one gets a kinetic equation that looks like this. So this is a partial differential equation where the variable we're solving for is the density of bacteria. Essentially, it's the same thing as the discrete model. Like the terms are all very, very similar. In particular, we have these interaction functionals, which are essentially like the Cucker-Smale interaction terms, where then we summed over all of the other bacteria. And here, because it's a continuous model, we integrate against the density. So essentially, it's the same thing, but packaged into one PDE. Nonlinear PDEs, we absolutely do not expect to be able to write down explicit solutions or even show that classical solutions exist. We hope to show weak solutions, which are like a mathematical formulation that's a little bit more general. I'm going to be a little sparse in the details here. I didn't prove existence of weak solutions. I proved existence of an even weaker thing called a measured valued solution. So we have a very wide class of objects that could potentially be solutions that are defined here. And then the theorem that I showed this summer is that for any initial condition with certain properties, there exists a solution with that initial condition that additionally satisfies a bunch of nice properties. The proof is a little bit in the weeds. So if you want to talk to me about it at the poster session or after this, whenever, I'd be happy to do so. So next, we're going to add stochasticity to the system. So this allows it to better model real-life situations where we might not know everything deterministically. We would like to add a Wiener process. That's the standard thing to make it stochastic, where we have a time-dependent, normally distributed noise. I am not a biologist, so I considered a bunch of places to add a random noise because I didn't know what would be most biologically motivated. You could consider a uniform drift on all of the bacteria. You could consider independent drifts on each of the bacteria. You could randomize the uptake. The one that I was particularly interested in was randomizing the communication. Our model supposes that every bacteria is able to perfectly know the position of all other bacteria. That seems a little bit silly. Here, for the sake of the presentation, I'll show the results for the first case, which is a uniform drift or uniform force on all of the bacteria, because that has the simplest result. So before we had an ordinary differential equation for each bacteria's velocity. Now we have a stochastic differential equation as highlighted by this dw term. To study this, we separate out the motion of the center of mass. The motion of the center of mass of all of the bacteria, which is the average velocity, from fluctuations from the average velocity. It turns out the reason this is so simple is that all of the randomness is trapped into the average velocity. And you could write out the stochastic differential equation, and that one is actually explicitly solvable. Here's the expected trajectory of the center of mass, as well as the variances of the trajectory and the velocity. Additionally, the fluctuations turn out to be entirely deterministic. Those don't have explicit solutions, but very easily simulatable and have very nice properties. These are just some numerical verifications that what we did was okay and correct. So on the left, every single center of mass trajectory is a random variable, and so we just take different realizations of it to see if the average of the realizations look like what we expected, and indeed it does. So every single colored line is a realization, and you see that the average and expected are pretty close. On the right, so that's just simulating the center of mass trajectory. This is just simulating the full system and showing that we didn't make an error separating out center of mass from fluctuations. We can see again, center of mass and expected are very close. The parameters of the system are just so everything goes up and to the right. Yes. So we see average and expected are very close. Note that I chose a very, very small sample size. 25 is not at all enough to verify the results are good. And also this takes not a lot of fractions of a second to run. I just did it so that all of the lines are individually visible. Additionally, 15 bacteria trajectories are a very small amount of bacteria. However, our system can handle much, much more. And so this is a first step towards an actual thing. So this is 625 simulations, and I wanted to verify that my variance calculation was correct. So you just take a large sample, you take the sample variance, and you show, hopefully, that is the same thing as theoretical variance, and indeed it is. All of the plots are estimators of the sample variance. So you can see the variance and estimators of the sample variance is kind of high. Additionally, I don't have it here, but I created a log-log plot to show that as you increase the number of realizations in log-log space, it's a linear line decreasing, which is what we expect. The slope is a little bit more than 1, which is a little bit mysterious to me for now, but I'm interested in exploring how that would work. In conclusion, some areas of future work are improving the modeling. The quorum sensing model we had in the beginning, it seemed a little bit arbitrary and contrived. A lot of the terms felt not super robust, and so I wanted to explore having more generic terms and to see if solutions were still tractable. Additionally, we wanted to construct more robust computational tools for simulating these things. Joshua Kalai has been working with me on that, or he's been working on it. Additionally, I was given a lot of free rein to kind of do whatever I wanted with the analysis. These are just areas that I wanted to explore, prove actual existence of weak solutions. We considered stochastic adjustments to the ODE version. We wanted to see how that will pass to the kinetic equation to get a stochastic mean field limit, prove better flocking results, develop a hydrodynamic formulation, which is we went from discrete bacteria to a continuous density bacteria to perhaps treating bacteria like a fluid and consider the moments. Oops. I would like to thank Jeffrey Kwan and Constantino Treviza for being excellent mentors on this project. I'd like to thank Joshua Kalai for helping me with this project understand quorum sensing. I'd like to especially thank Daniel Serrano for being an excellent program coordinator. This is a very difficult summer for everyone. So him being a very good coordinator made this summer very nice. I'd like to thank the NSF. Cool. I would like to thank the NSF for funding this REU program. I have some references. There's plenty more. Thank you all for attending my talk. I have questions.